Morning, friends. Today we are going to discuss the first topic of Science and Technology, Part 2, Heredity and Evolution. Now, in this topic, we are going to discuss the hereditary and hereditary changes, transcription, translation, and translocation. Evolution, evidences of evolution, Darwin's theory of natural selection, Lamarckism, speciation, and human evolution. Friends, let us understand what do you mean by heredity. Now, heredity you have learned in ninth standard regarding heredity. It is nothing but transfer of characteristics from parents to the next generation. What are being transferred and who is going to transfer these characteristics? Now, here the characteristics, the transfer of characters or traits from the parents to their offsprings is called as hereditary. The transfer of biological characters from one generation to another via genes. Now, what exactly is getting transferred? What characters they will get transferred? That we all know the characters when they are being getting transferred. Now, which characters will get transferred? The physical appearance, the phenotypic characters, and the genotypic means genetic constitution. The physical appearance, the color of the skin, color of the hairs, height, all these factors which we see, they are being transferred from parents to the next generation. But how they are getting transferred? They will get transferred by an important segment called as genes. Now, what is genes? Gene is nothing but a functional segment of DNA. So, who is responsible for transfer of characters? We say the DNA is getting copied. So, DNA, what is DNA? DNA is nothing but a nucleic acid which is present in the nucleus of an cell. Now, the full form of DNA is deoxyribonucleic acid. If you see the structure of DNA, it is double helix structure. Double helix structure, it was given by the scientists Watson and Crick, and they were awarded with the Nobel Prize in 1953 for their double helical structure of DNA. This DNA, the part of DNA which is of our use or which is helping us in the functioning, which has a particular protein which is required, that is called as gene. So we say gene is functional segment of DNA. Now, where these genes they are present in the DNA, no doubt, but how they are copied from one generation to other generation, how they transfer from one generation to other generation, that is by chromosomes. Now, we have studied last year different types of chromosomes, how actually the DNA is a double helical structure, but when it is properly coiled, DNA when it is properly coiled, it will be paired in the form of chromosomes, and these chromosomes they get transferred from one generation to another generation. We know in human beings there are 23 pairs of chromosomes, that is 22 pairs they are autosomes and one pair is a sex chromosome, that we have studied last year. Now let us understand first the structure of DNA. In the DNA it has phosphoric acid, then a deoxyribose sugar, that is a pentose sugar, and a nitrogen base. Now, what are the types of nitrogen bases? Nitrogen bases, there are two types actually, purines and pyrimidines. Purines are of two, that is adenine and guanine, whereas pyridine, if you see, that is cytosine, and in certain case in RNA it is uracil. Now, this phosphoric acid molecule, it is attached to the fifth number carbon. If you see the structure of sugar, it is a pentose sugar. See here 1, 2, 3, 4 and this fifth carbon. This fifth carbon is attached to a phosphoric acid molecule and the first carbon is attached to a nitrogen base. Now, the second carbon, when this second carbon, the oxygen is absent at the second carbon, we call it as deoxy, that is absence of oxygen at the second carbon. So it is called as deoxyribose sugar. And in case of RNA, there is a ribose sugar. If you see the structure of ribose sugar, here the structure, first carbon will have a nitrogen base, second carbon will have hydroxyl group, third carbon also will have hydroxyl group, and the fifth carbon will be attached to a phosphoric acid. So in DNA, what is there? There is phosphoric acid, deoxyribose sugar, and a nitrogen base. In RNA, there will be a ribose sugar, phosphoric acid, ribose sugar, and the nitrogen base. Now, you might have understood that nitrogen bases, there are two types, purines and pyrimidines. There is also this one, purines always pairs with the pyrimidines. Now, in the pairing, you will see that adenine always pairs with thymine, whereas guanine pairs with cytosine. Now, the bonds which are present here, adenine and thymine, there is a double bond, whereas in guanine pairs with cytosine with a triple bond. In case of RNA, instead of thymine, the thymine is replaced by uracil. So, uracil will be present in case of RNA. Now, only DNA is responsible for transfer of characteristics. In exceptional cases, if you see in the case of virus, you will see that the virus, it is all other animal, that is it is DNA is responsible. In case of virus, you see virus is a microorganism which is non-living outside the body, but when it enters our body, it gets the characteristics or it gets the material which help us it to replicate. It starts replicating and it becomes, we can say, living when it enters the body. So virus is a exceptional case. Now what actually is evolution? See, primitive organisms, the first organisms which are present on the earth, they were called as prokaryotes. Then unicellular organism, where only a single cell was responsible for carrying out all the functions of the body. We know unicellular organisms like amoeba, euglena, but when there was a further gradual change, it get converted when the nucleus was covered with a nuclear membrane. All the cell organelles were covered with membranes, cell membranes and the membranes. The new cell which was advanced cell which is present now, that is eukaryotic cell, and these organisms were called as eukaryotes. Further it got converted multicellular. Now what actually is evolution? It is a gradual change in living organism. It is, we say, we are evolved from monkeys. Homo sapiens, our species is what? Homo sapiens, that is human beings. They are being evolved from monkeys. So there is a step by step process. Evolution, it is a gradual change in the living organism. It is a slow process and it leads to drastic changes after some time. So this is a step by step process which help us to have the required changes in the living organism, which helps the living organism to sustain in that particular climatic conditions, in that particular situations. In this topic we are going to discuss Darwin's theory also, that how nature select the variety of the species. Now friends, how actually it went on? Now see, let us see again the hereditary transfer of biological characters from one generation to another via genes. Now you might have understood that what is genes? Genes is nothing but a functional segment of DNA. Why it is called as functional segment? Because it helps us to, it consists of required protein molecule which will help us to get transfer. How they will be transferred? Via chromosomes. So chromosomes will be copied, enter, we can say they get transfer from one generation to another. Now who is the father of the genetics? The scientist John Gregor Mendel. Last year we have discussed the monohybrid ratio and dihybrid ratio and how genetics came into existence. So John Gregor Mendel, he has done a valuable work, good research in the genetics, and he is the pioneer in the field of genetics. So he is called as father of modern genetics. Then further we say there is variations. Now why variations are there? In asexual reproduction, if you see a plant of rose look like a rose. But are we human beings all in sexual reproductions? If you see, even twins are also not similar. Why? Because there are variations. Variations lead to the changes which are there seen. Exactly, a child looks like some characteristics are of parents, but there are certain characteristics which are differing. That is due to variation. And variation is what? It is a product of sexual reproduction. So, in 1901, a mutational theory explained that it leads to the variation. Now, in 19, if you see the history, in 1902, Water and Sutton he gave that they have seen paired chromosomes in the grasshopper cell. How is the nature of genetic material? That was proved that genes are being carried from one generation to other via chromosomes. So I told you just now that chromosomes they will be transferred from one generation to another. Then in 1944 scientists proved that except virus all living organisms have DNA as a genetic material. Genetic material means which is responsible for carrying the hereditary characters. In 1961, you see the French genetist proposed. What is genetist? The one who study regarding the genetics. He proposed a model for protein synthesis with the help of DNA in a bacterial cell, and then the recombinant technology came into existence. Now friends, this is just a platform which we are trying to prepare for learning this topic. Let us see what are the basic questions which are being asked in this topic at the starting. See, which component of the cellular nucleus of living organism carries hereditary characters? I hope you might have understood the answer is DNA. Which component who will be carrying the information hereditary characters? That is DNA. Full form is what? Deoxyribonucleic acid. What do we call to the process of transfer of physical and mental characters from parents to the progeny? Progeny means offsprings, or we say next generation. That process or that biological process is called as hereditary. Now what are the components? Which are the components of DNA molecule? I told you just now DNA molecule is made up of three basic components. What are they? First is phosphoric acid, then a deoxyribose sugar and a nitrogen base. Nitrogen base, that is purines and pyrimidines. So phosphoric acid, deoxyribose sugar and nitrogen bases. Why it is deoxyribose sugar? Because it is there is absence of oxygen on the second number carbon. Ribose sugar means a sugar, it is a 5 carbon sugar. It is a 5 carbon sugar. So the science of hereditary and is useful for diagnosis, treatment and prevention of hereditary disorders, production of hybrid varieties of plants and animals in industrial process, and also everywhere you can see in our day-to-day life, genetics plays a very important role, and there is a vast scope in the field of genetic engineering. So this I am going to add a video which will help you to understand the process, that is transcription, translation and translocation, which we are going to see in the next session. Please listen to the video, go through the video clearly and sincerely, so that you will able to get the concepts of basic concepts of this transcription, translation and translocation very easily in the next session.
Transcription, translation and translocation. Proteins are needed for carrying out various body processes. Here in this video, we will understand the process of protein synthesis, which involve transcription, translation and translocation. Ideally speaking, proteins are synthesized in the cell of the body. The information needed for protein synthesis is present in the deoxyribonucleic acid or DNA of the cell. It is located inside the nucleus. However, the actual process of protein synthesis takes place outside the nucleus. The information from the DNA is transcribed or copied to messenger ribonucleic acid, a molecule similar to DNA but with single strand. It in turn transports this information to ribosomes. The ribosomes and transfer ribonucleic acid use this information to produce the proteins. This basic flow of information from DNA to RNA and to ribosomes is called the central dogma in science. For understanding it in detail, let's first revise the knowledge about the DNA. A DNA is a long molecule consisting of two strands that form helical structure like a twisted ladder. This structure is known as double helix. DNA is a cookbook of the cell which consists of thousands of small segments, some of which stores the recipes to produce proteins. Such segments are known as genes. It is made up of sugar phosphate backbones with pairs of nitrogenous bases. Adenine always form pair with thymine, and likewise cytosine always form pair with guanine. The basic structural unit of DNA consists of phosphate group, 5-carbon sugar and one of the 4 nitrogenous base and is called as a nucleotide. With this basic knowledge, let's understand the 3 processes. Initially, an enzyme called RNA polymerase acts on DNA and unwind it from the starting sequence to be transcribed or simply copied. The enzyme takes only one strand as template and produces its complementary strand. The unusual thing here is the formation of base uracil instead of thymine. It happens because formation of uracil is energetically less expensive. The most important thing is information remains same even if the format is changed. It is just like noting down the recipe from cookbook in your own words. The information is same, but the format is different. Once the transcription process is completed, the DNA strands rejoins and the mRNA is transported outside the nucleus into the cytoplasm. The ribosomes gather around the mRNA and here starts the process of translation. Understand it like producing the food from the recipe. The 3 nucleotides on mRNA forms a code for an amino acid. These are called triplet codon. Amino acids are the basic building block of proteins. For every triplet codon, there is transfer RNA or tRNA carrying an amino acid. The sequence on tRNA is called anticodon. Inside the ribosome, the start codon is recognized. A tRNA having anticodon with complementary sequence attaches to it. Likewise, the next tRNA attaches to next triplet codon. The ribosomal ribonucleic acid or rRNA present in ribosome binds the amino acid brought by the 2 tRNAs with peptide bonds. Once the bonding is done, the tRNA corresponding to first codon transfers amino acid to tRNA corresponding to next codon. This whole process is termed as translation. The ribosome moves in forward direction on mRNA by a distance of 1 triplet codon so as to repeat the translation. This is called translocation. As a result of transcription, translation and translocation, there is formation of an amino acid chain. The chains formed by different ribosomes joins together so as to form a complex protein. Together, these processes can be termed as gene expression. The familiar example is transfer of characters from parent to child. Sometimes sudden changes occurs in gene. Sometimes any nucleotide changes its position. It causes a change in the organism's character. This is called mutation. I hope you have understood the concept of transcription, translation and translocation very well.
Thank you, dear friends.